the pill.
Welcome to the Deep Dive. We're here to really get into the weeds on complex topics and pull out the key insights for you. And today, it's a really fascinating and I think critical area, human drug metabolism. Now, this one's special. We're tailoring this deep dive specifically for you, thinking about your PhD qualifying exam. You shared a chapter on biosynthesis and drug metabolism, and well, our mission is to break it all down. We want to give you explanations that are simple, easy to remember, basically lecture ready, with real-world examples too. The idea is you'll walk away feeling really solid explaining this stuff yourself.
Exactly. We're aiming for that crystal clear understanding of how the body handles drugs. Absorption, distribution, transformation, elimination. The whole journey. It's always about finding that balance, isn't it? Keeping drugs effective, but more importantly, keeping them safe.
Okay, so let's start right there with what seems like the basic challenge of giving someone a drug. You're targeting maybe just a small area, some specific cells, but you end up dosing the whole body, right? That feels like an immediate problem.
Oh, it absolutely is. It's a fundamental compromise you have to make. It's like a constant tightrope walk, really, between the good effects you want and, well, the potential side effects everywhere else. I mean, drug companies work incredibly hard to screen out drugs with really bad side effects. So the ones that make it to market, they generally have manageable side effects. But, and this is key, usually only within a very specific concentration range.
Right, that specific concentration range. That sounds crucial. Is there like a formal name for that ideal zone, that sweet spot?
Yes, there is. We call it the therapeutic window.
Yeah.
If you look at figure 1.1 in your chapter, it illustrates this nicely. It's basically that range of drug concentration where you get the best pharmacological action with the fewest side effects. For something simple like, say, paracetamol for a headache, you might just need to be in that window briefly. But think about drugs for chronic conditions like high blood pressure or thyroid issues. You're talking repeated doses, days, months, sometimes years. That's a whole different ballgame.
And I imagine over those long periods, the concentration levels wouldn't stay perfectly stable. Things like being ill or what you eat or maybe other medicines, they could push the levels around.
Precisely. That's a huge factor. If the drug concentration dips below that therapeutic window, it's simple. The drug fails.
Yeah.
It won't work effectively. But if it climbs above the window, well, then you start seeing intensified actions, both the intended ones and, more worryingly, the off-target adverse effects. And that can escalate pretty quickly to stress, incapacitation, sometimes, sadly, even death. And here's something really vital for you to remember for your exam. Everyone's therapeutic window is a bit different. There's huge individual variation. It's never one size fits all.
Okay, so the window is the practical safe zone. But how do scientists or doctors measure drug safety more, formally, especially when comparing drugs or developing new ones? Is there a number?
Yes, and that number is the therapeutic index, or TI. And the beauty of the TI, really, is that it gives you a quick snapshot, sort of an at-a-glance idea of how much wiggle room you have before a drug turns dangerous. It's defined simply as the ratio between the toxic or lethal dose and the dose that's effective. So take drugs with a narrow TI, things like lithium, or maybe those older barbiturates from the 60s. Their effective dose and their toxic dose might only be twofold apart, sometimes even less. That means a tiny increase in dose or just a bit of accumulation in the body can flip you from therapeutic to toxic very, very easily.
Which explains why we've seen such big shifts in treating things like depression, right? The older drugs, the TCAs.
The tricyclic antidepressants, yeah. They had narrower TIs, which made overdoses a really serious risk.
Exactly. Newer drugs, like the SSRI's selective serotonin reuptake inhibitors, they generally have much wider TIs. That's a massive step forward for safety, significantly reducing that overdose danger. But it's important to note, many drugs with narrow TIs are still very much in use today. Phenytoin, carbamazepine, valproate for epilepsy, warfarin as an anticoagulant. For these drugs, you have to be much more vigilant because accumulation can have severe consequences and often much faster than with wide TI drugs. The stakes are just higher.
So missing that sweet spot, that therapeutic window, what does that actually look like in a clinical setting? What are the sort of the two big dangers if drug levels go wrong?
That's a great way to frame it for your exam. You essentially face two major pitfalls. Drug failure on one side and drug toxicity on the other. Your figure 1.2 lays this out. Should we start with drug failure?
Yeah, let's do that. Well, it leads to drug failure. It's not just that the drug isn't inherently good. Right. Drug failure happens when a drug, even one that should work, just doesn't reach high enough concentrations where it needs to act either in the general circulation or at the specific target cells. Why? Well, maybe it's poorly absorbed from the gut. Maybe it gets bound up somewhere else in the body. Or maybe it's just removed too quickly. A really common scenario, something you'll definitely see, is when you introduce a second drug that speeds up the removal of the first one. Suddenly, the first drug's levels just plummet, and the clinical results can be serious. For someone with epilepsy, it could mean losing seizure control, risking injury. For contraceptives, it could mean an unwanted pregnancy. For antipsychotics, maybe relapse and hospitalization.
And you mentioned a wider impact too, like with antibiotics.
Absolutely. With antibiotics or antiparasitics, if their levels drop too low because of rapid clearance below what we call the minimum inhibitory concentration, you risk selecting for drug-resistant bugs. That's a huge public health issue. A key point to remember here, maybe jot this down, is that drug failure usually happens gradually. It can take many days before it becomes obvious.
Okay.
So now let's look at the flip side, drug toxicity. This is when the drug accumulates. Maybe the dose was too high to begin with, or very often the body's removal systems fail. And frequently, that failure is caused by another drug interfering with clearance pathways. The way it unfolds can be quite insidious. First, you might just see an exaggeration of the drug's intended effect. Then you start seeing off-target effects pop up. And in the worst cases, you can get irreversible damage to tissues or organs.
And we've seen some really serious examples of that, unfortunately.
We have cyclosporine, which is used after transplants. If it builds up, it can cause severe kidney damage, even organ failure. Or think about anticonvulsants or antipsychotics. Too much can lead to real confusion, extreme drowsiness. And a historical example, maybe one to mention, is the antihistamine terfenadine. It was pulled from the market because if it accumulated, usually due to interactions, it could cause fatal heart rhythm problems. And here's the critical difference compared to drug failure. Toxicity can happen much, much faster, often within hours, not days. Rapid intervention is absolutely vital.
Okay, so we've got the stakes, clear failure, or toxicity. This leads us naturally to how the body actually gets rid of these drugs, the cleanup process. What's the term for that?
That whole process falls under the umbrella of clearance. It's generally defined as the removal of drug by all processes from the biological system. Or, maybe a more technical way to think about it, it's the volume of fluid, plasma, blood, whatever, from which the drug is irreversibly removed per unit of time. Now, it's not like the drug just vanishes, poof. Often, removal involves biotransformation, which is just metabolism changing the drug's chemical structure. And usually this change reduces or gets rid of the drug's therapeutic effect.
But not always, right? You mentioned prodrugs.
Exactly. Good recall. A prodrug, like clopidogrel for blood thinning, is actually inactive until it's metabolized. That's the exception. But generally, the metabolites that transform products are removed faster and end up being excreted, usually in urine or feces. Though again, there are always nuances. Some metabolites might still be active, like disipronine or morphine 6-glucuronide. And sometimes metabolism can actually make something less water-soluble, harder to excrete, like with acetylated sulfonamides.
So metabolism changes the drug, but for actual physical removal from the body, who does the heavy lifting?
Okay, so the term for physical removal is elimination. And the main players there are your kidneys. They are fantastic at filtering out smaller, water-soluble chemicals from the blood. They filter a huge volume constantly, but they have a significant limitation. They're not very good at eliminating lipophilic chemicals, those oil-loving or fat-soluble ones. Why? Because even if a lipophilic drug gets filtered by the kidney, it tends to just diffuse back into the bloodstream from the kidney tubules, because the kidney tissue itself is, well, lipophilic.
Ah, okay. So those lipophilic drugs are tricky. How does the body deal with them then if the kidneys can't easily just filter them out?
This is where biotransformation prior to elimination becomes absolutely critical. Those lipophilic drugs have to be chemically changed, made more water-soluble, so the kidneys can then effectively eliminate them. The main organs doing this biotransformation are the liver, the gut, the kidneys themselves, and the lungs. They basically grab the drug, chemically modify it, and then often release the now water-soluble metabolite back into the blood where the kidneys can finally handle it. The liver is the major powerhouse here. It can also excrete some larger drugs and metabolites directly into bile, which then leaves the body via the feces. And we shouldn't underestimate the gut microbiota either. Those bacteria in your gut can have really profound effects on how drugs are handled. That's a growing area of research. Now, related to this is a concept called intrinsic clearance. Think of this as the inherent sort of raw power of an organ's metabolic machinery to transform a specific drug. It's measured independent of things like how fast blood is flowing through the organ or how much drug is bound to proteins. It's about the enzymes themselves. Of course, the actual clearance by an organ depends on many things. How big the organ is, blood flow to it, the total amount of those biotransforming enzymes, their intrinsic clearance for that specific drug, protein binding, and the drug's own lipophilicity. And when we talk about total body clearance, call total, it's simply adding up the clearance from all the individual organs, liver, kidney, gut, lung, etc. For example, a drug like atenolol is almost entirely cleared by the kidneys. Paracetamol uses both the liver and kidneys. Ethanol alcohol uses liver, kidney, and lungs.
Got it. Now, there's this other concept that sounds really important for oral drugs, the first pass. You use that analogy of trying to get into a castle by knocking on the front gate. Can you unpack that a bit? What is this first pass effect?
It's a great analogy because it highlights the barrier. Your figure 1.3 shows this well. First pass basically refers to all the metabolic processes that jump on an orally taken drug before it even gets into your main systemic circulation. A significant chunk of the dose you swallow can get metabolized, essentially chewed up and inactivated, primarily by the gut wall and the liver, before it ever reaches the bloodstream to do its job.
And you mentioned the gut plays a surprisingly big role here. It often seems like the liver gets all the attention from metabolism. Why is the gut so significant in this first pass?
That's a really important point and often overlooked. When you swallow a pill, it first encounters the cells lining your gut, the enterocytes. Once the drug gets inside these cells, a couple of things happen. First, these cells have their own set of drug metabolizing enzymes, quite similar to the liver's. Second, they have efflux pumps, proteins that actively push drug molecules back out into the gut lumen. This back and forth, combined with the metabolism happening right there in the gut wall, means the gut makes a really substantial contribution to first pass loss, even though its total mass of enzymes is less than the liver's. Why is it so effective? Probably because the drug concentration is very high right there locally. The enzymes might have high intrinsic clearance for certain drugs. The drug spends a fair amount of time transiting the gut, and you don't have the limitations of blood flow or protein binding that you see in the liver. We see this with drugs like midazolam or cyclosporine. Maybe up to half the oral dose can be knocked out by the gut before the drug even sees the liver. It's a major gatekeeper.
Wow, okay. So after potentially losing a chump in the gut, the drug that survives travels, I assume via the portal vein, directly to the liver, and the liver takes another crack at it. How do we measure the liver's contribution?
Exactly. What survives the gut goes straight to the liver, which gets the next shot. We measure the liver's efficiency here using hepatic extraction, E. Simply put, E is the fraction of the drug entering the liver that gets removed or metabolized during that single pass through it. It's often expressed as 1 minus the fraction that escapes. So if 70% is metabolized, E is 0.7. Clinically, we often categorize drugs based on this. High extraction drugs have an E greater than 0.7, low extraction drugs less than 0.3, and then there's an intermediate category. Let's talk about high extraction drugs first. For these, the liver is incredibly efficient at removing them. The processing is rapid. Either the enzyme systems are very abundant, or maybe the drug looks a lot like something the liver normally processes in large amounts. A good example for you might be AZT, zitavudine, it's an anti-HIV drug, looks like thymidine, and the liver clears it incredibly fast, giving it a very short half-life, like an hour or less. For these high extraction drugs, the intrinsic clearance, the enzyme capacity, is so high that it's not the limiting factor. The real bottleneck is just how fast the drug can be delivered to the liver via blood flow. So hepatic clearance essentially equals liver blood flow. There's hepatic Q.
So that's the efficient toll booth analogy again. The enzymes are ready, they just need the cars, the drug molecules, to arrive faster.
Precisely. And interestingly, for these drugs, protein binding isn't really a barrier either. The liver's extraction process is so efficient it can strip the drug off the binding proteins. We call this unrestricted clearance. Examples: pethidine, metoprolol, propranolol, lignocaine, niftecaman, fentanyl, verapamil. Quite a few common ones. And the clinical relevance is huge. If liver blood flow changes may be due to aging, liver disease, heart failure, it significantly impacts the clearance of these drugs. Slow down blood flow, you slow down their removal, potentially leading to accumulation. Okay, now let's shift to low extraction drugs. As the name implies, the liver clears these much more slowly. Maybe the enzymes struggle to metabolize the drug's structure, maybe there isn't much enzyme capacity, or maybe the enzyme levels are just low. These are often called low intrinsic clearance drugs. Now, if a low extraction drug is not heavily bound to proteins, say less than 50% bound, its clearance is pretty much directly proportional to that intrinsic clearance of the raw enzyme efficiency. But, and this is a key distinction for your exam, if the drug is highly protein bound, think phenytoin or valproate, around 90% bound, then the clearance gets hampered. Why? Because the liver enzymes have a lower affinity for the drug than the binding proteins do. The proteins hold onto it tightly.
So it's not just the toll booth efficiency, but also how much drug is actually free and available to even get to the toll booth. The protein binding acts like traffic congestion.
That's a perfect way to think about it. The clearance depends not just on the enzyme's intrinsic ability, clinintrinsic, but also on the fraction of drug that's actually unbound and available. So the formula becomes clopatic, clinintrinsic Fu. The more it's bound, the lower Fu, the slower the overall clearance, even if the enzymes themselves are reasonably efficient. We sometimes call this restrictive clearance because the binding restricts access to the enzymes.
So for these low extraction drugs, blood flow isn't the main driver, but enzyme activity and protein binding are.
Exactly. Changes in liver blood flow don't have much effect, but changes in intrinsic clearance. Maybe another drug inhibits the enzyme or induces it, making it work faster. Those changes significantly affect the levels of the pharmacologically active free drug. This often means you need to adjust the dose. And here's another subtlety. For those highly bound, low extraction drugs, if something causes protein binding to decrease, maybe kidney problems leading to lower albumin, the total drug level might fall. But the free drug level, the bit that matters for effect and toxicity, often doesn't change much. So interestingly, you might not need to adjust the dose in that specific situation. Diazepam, naproxen, metronidazole are examples here.
Wow, okay, so you've got the gut taking a piece, the liver taking potentially another large piece. All this first pass activity must dramatically affect how much of an oral dose actually gets into the system, right? Which brings us to bioavailability.
Absolutely. All those hurdles sum up to determine absolute bioavailability, which we usually just call FF. Tells you, quite simply, what fraction of an oral dose actually reaches the systemic circulation compared to the same dose given intravenously, which is 100% bioavailable by definition. Mathematically, you can think of F as a product of three fractions. F, the fraction absorbed across the gut wall into the enterocytes, Fg, the fraction surviving metabolism in the gut wall, and Fh, the fraction surviving metabolism during that first pass of the liver. So F is Fa, Fg, Fh. Now, if a drug is subject to high first pass extraction, either in the gut or liver or both, it will likely have poor bioavailability. An F of, say, 0.2 means only 20% of the oral dose makes it into circulation intact. This means you need a much bigger oral dose than an IV dose to get the same effect 5 times bigger in the F.2 example. And that difference, that reliance on getting through the first pass, makes these drugs inherently more variable and potentially more problematic in clinical use. Let's really try and drive home the consequences of changes in clearance on plasma levels, especially for these tricky drugs. Imagine a worst-case scenario. Drug X. It has poor bioavailability, let's stick with X0.2, and has a narrow therapeutic index. Very dangerous combination. Let's say the safe and effective IV dose is 20 milligrams, because F0.2, the standard oral dose would have to be 100 milligrams to achieve that same systemic exposure. Now, what if something changes? What if the first pass effect is suddenly reduced? Maybe another drug inhibits the gut or liver enzymes. Suddenly, instead of 20 milligram equivalent reaching the plasma from that 100 milligram pill, maybe 50 milligrams or even the full 100 milligram gets through. The plasma levels would skyrocket, potentially 3, 4, or 5 times higher than intended. For a narrow TI drug, that's almost certainly going to cause severe toxicity, maybe even death. Or flip it the other way. What if clearance is suddenly accelerated, maybe by an inducing drug? Now, perhaps effectively none of that 100 milligram oral dose makes it into the plasma. You get complete lack of efficacy. Drug failure. It just highlights how critical understanding metabolism and clearance is, and how sensitive drug effects can be to changes in these processes. The stakes are incredibly high.
That really puts it into perspective. This whole deep dive, it's underscored how sophisticated the body's systems are for handling foreign chemicals. We've covered the therapeutic window and index, the dangers when concentrations shift, and then these really complex processes of clearance, biotransformation, and that crucial first pass effect.
It does make you think, doesn't it? What did these systems originally evolve for? Because it certainly wasn't for handling modern pharmaceuticals. We're talking about ancient, complex, highly adaptable machinery. Its original job was likely protecting the organism from naturally occurring toxins in food, controlling the levels of our own signaling molecules, hormones, things like that, and performing all sorts of other physiological tasks. Our drugs are just, well, newcomers tapping into this very old system.
And hopefully this discussion gives you a really solid foundation. You should feel well equipped now to explain these core ideas, almost like you're giving the lecture yourself for that PhD exam. You've got the concepts, the examples, the clinical relevance.
Absolutely. And just to help you lock it in, maybe test yourself with these few questions. Think about how you'd answer them clearly and concisely, like in an exam setting. First, explain the concept of the therapeutic window and the therapeutic index. Give me a real-world drug example for each and explain why these concepts are clinically significant. Second, describe the first pass effect.
Form a pill.